Okay, so chapter 4 specifically talks about obviously DNA, RNA, nucleotides, nucleic acids. The main point to kind of understand is that when you talk about monomers, right, of DNA, when you're trying to make DNA, how do you make DNA? Just out of curiosity. And don't worry, by the way, if you don't know the answer. And it's okay if you don't write notes either. Just, we're talking to each other, okay? Don't worry. So basically, the way to make DNA, okay, it starts off with nucleic acids, yes.
Okay.
That's the polymer, okay?
Yeah.
But the monomer, so you go nucleic acids, okay? To get to nucleic acids, though, you need nucleotides, okay?
Attach to this, attach to that. Methyl group, attach to this, attach to this.
Nucleotides are really important because when you start to stack them together, when you start to connect them through phosphodiester linkages, I need it. So when you're trying to make those nucleic acids, you're just taking nucleotides, bonding, nucleic acids, and then from there, what do you get? Get your DNA helix, right? So the thing though about nucleotides is that you have different components of it, right? Do you know the 3 components of it?
Yes.
What component?
Huh?
What component?
The components of a nucleotide would be
The phosphate sugar.
Exactly.
The base.
uh-huh.
The base.
Uh-huh.
The 5 sugar.
Yeah, exactly, right? So you're talking about the pentose sugar.
Yeah.
You're talking about the phosphate,
The
right?
bases.
And then you're talking about the nitrogen base, right? So you don't have to know this right now, but when you're in biochem, you're going to have to know the structure of it. So if we're talking about the structure, we're looking at something like this. Ready? So this is kind of interesting, right? Nitrogen base, you have your sugar, okay? And then you have your phosphate. I didn't write out the phosphate, but if I want to write it out, I just write it like this.
Can't give up after 15 minutes. That's too early. An hour 45 maybe.
And this has 2 phosphates. Sometimes you can put 3 phosphates.
Okay.
So let me give you a really good example. If we're talking about ATP, right? You talk about adenine, triphosphate, exactly, right? So your nitrogen base would be an adenine, right? And then, or adenosine, adenine, I usually pronounce those incorrectly. But if I were to ask you, is it a purine or is it a pyrimidine, what would you say? Purine. Yeah, exactly. What's the main aspect that you should know about a purine?
Maybe thymine is replaced or something like that, or like double bonds or triple bonds.
Oh, that's smart. You're talking about the base pairing. What you should know though about purines, if we're talking about structure, we're talking about the fact that it has 2 fused rings. 2 fused. So I'm not going to draw this correctly, but if I were to draw like adenine or something, you see how it has 2 fused rings, right? Versus a purine might just only have 1 ring. That's the main component of purine versus a pyrimidine. But yeah, you're right about the whole double bond, or not double bonds, but you're talking about hydrogen bonding between adenine and thymine, and then cytosine and guanine. That's really important. And in the future, because you mentioned your pre-med and stuff, you're gonna see, like, in future exams, right, your medical college admissions test, they might ask you, which bond is more stronger? Something with a lot of CGs in it or a lot of ATs in it? What do you think? If something has more CG bonds in it versus more AT bonds, what do you think is going to be stronger?
The one that has more bonds, like the double bonds.
Yes, ma'am. So when we talk about double bonds, you mean hydrogen bonding, right? So we're talking about something like that, right? But we know that A and T usually has 2 hydrogen bonds to each other. If we're talking about C and G, we're talking about 3.
Yeah.
So if we have more CGs, that means that it requires more energy, more heat to break something which has a lot of CGs in it because of those hydrogen bondings, right? So that's kind of, like a little introduction. If we're talking about nucleosides though, what's the difference between a nucleoside and a nucleotide? Ooh, think. Just tell me. There's one thing that's different from a nucleoside versus a nucleotide. It doesn't have a phosphate. So with nucleosides, you're just gonna have the pentose sugar and you're only gonna have the nitrogen base. You basically lose this side, okay? That's kind of the main aspect of nucleoside versus a nucleotide. Give me 1 second, I wanna log in, make sure we stay on track because I don't have a PowerPoint or anything like that. So, now you get it, right? The next thing that I kind of want to talk about is, the functions of nucleotides, right? So we mentioned that there's ATP, right? ATP, where do you think you usually see that? ATP. If I were to ask you, where would you find ATP? I'd tell you that it's probably currency, energy currency, right? So like, in my body, if, let's say I'm running a marathon, and I take lots of carbohydrates in, you're gonna learn this soon, you do glycolysis. Glycolysis means we're gonna break down those sugar and we're gonna use it for energy. Let's go run, right? We're gonna break down the sugars and use it for energy. The energy that you yield would be ATP, right? So you can see that a nucleotide, for example, like ATP, which has the adenine and 3-phosphate, I know there's only 2 here, and then you have your pentose sugar, would typically be something for energy currency, metabolism, things like that. That's where you would see ATP. Now, on the other hand, there's another thing called GTP. GTP, what do you think the difference is? Just a nucleotide, right? Instead of adenine triphosphate, it's guanosine triphosphate, right? So you just have the guanine here. Usually, you see GTP for protein transfers. So, you're gonna learn this soon. But when you want to signal something in your body, okay? I'll give you another really good example. Have you heard of epinephrine? Have you heard of adrenaline, right?
Yeah.
Have you heard of fight or flight? If I want to fight or flight. So let's say somebody is, okay, I want to make sure this is like PG. I mean, it's not PG. I mean, you're like, what, like 21 or something, 18, okay? So let's say that you're trying to run away from somebody. It's a tiger. A tiger's coming after you, right? You're either going to want to fight the tiger or you're going to run away from the tiger, okay? In that case, we're going to see adrenaline, epinephrine, this thing called a signal transduction pathway. You're going to learn this later on in the class. And what's going to happen here is that the adrenaline, okay, which we call essentially epinephrine, I'm just going to erase this so I can show you what I'm talking about and why we talk about GTP, because GTP is very, very important. You're going to see this in the future 100%. So if this is, have you heard of a phospholipid bilayer?
Yep.
Yeah, okay. On our phospholipid bilayer, we have these things called receptors. You're gonna learn this soon. It's towards the end, exam 4, for a cell bio. So you're gonna have like this thing called epinephrine, okay? It's gonna come onto the receptor. What's gonna happen here is we use this thing, it's a G-coupled protein receptor, don't need to know that right now, but you're gonna see like GTP, the energy, is gonna be used to move certain elements inside the cell so that you can either run away or you can fight the tiger. So do you see what I'm saying? You're using GTP as energy to move the signal that came in your body so that you can run away or you can fight it. See what I'm saying? So it's still energy, it's just used in a different method. So that's kind of that's what you usually see, ATP, GTP. Of course, you see this other thing called cyclic AMP. I'm not gonna go over that. It kind of has to do with that as well, okay? So now, the next thing I'm gonna talk about is linking nucleotides. So you know how I told you that you have to link these nucleotides so that you can get nucleic acids, right? And then when you get nucleic acids, you can make this huge DNA helix, right? So the thing is though, is that in chapter 3, you kind of learned this, condensation reactions. Do you know what a condensation reaction is?
In which we, like, the hydroxide ion is released, like water ion is released.
What's the objective? Ooh, this is really ugly. The objective, like if I'm telling you, why do we do condensation reactions? What would be your answer? Typically, the condensation reaction is meant to build something. It's to build something, right? We want to make bonds, right? So we want to make bonds, okay? You don't want to break bonds. That's not what a condensation reaction does. What it does is that we bring one nucleotide A, this is nucleotide A, we bring it with nucleotide B, so another nucleotide, let's make it T or something, I don't know, you get my point, right? You bring one nucleotide and another nucleotide, when you link them together, yeah, you're right, water does leave. That's 100% correct, water does leave. But the idea here is that now you just linked two nucleotides together. You need to know condensation reactions. Listen, condensation reactions are everywhere. They're in nucleotides, they're in carbohydrates, they're in proteins, right? Making every single bond. Glycosidic linkages, peptide linkages, these are phosphodiester linkages, okay? Do you know what I mean when I say phosphodiester linkage? Like which aspect of it?
Yeah, everyone's clear. I just like.
No, don't worry, don't worry, don't worry. I'm telling you, right? The phosphodiester linkage is talking about basically you're linking at the phosphates. So essentially what am I trying to tell you is, if I have a nucleotide, okay? And I'm gonna draw my nucleotide one more time so you can see what I'm talking about. Okay? OH, I have my nitrogen base, we're gonna call it, let's call it thymine, okay? That's the nitrogen base up here, okay? And we'll have CH2, and then we're gonna put the phosphate here, okay? Okay, so that's my this is my nucleotide, okay? When I wanna link this to something else, okay? If I wanna make a chain, okay? I'm not gonna show you the reaction for it because it is a bit confusing, but I can show you where the water comes from, okay? Bear with me. So, usually, if you want to link, it's a very messy kind of thing because in your unit you don't learn how to link it together. You just learn what they're bonded by, right? You just say, oh yeah, it's linked by phosphate, whatever. So that's why I kind of don't want to go through how you do it because that's more of a biochemistry mechanism. But you have to know 5 to 3. I don't know if you saw that in the slides, but she talks about if you want to build nucleotides, if you want to continuously keep adding, like I told you, right? Because you're making nucleic acids, you need to add from the bottom up. So what am I saying?
It moves from 5 to 3 direction, and the 3 of the previous one, the previous strand attaches to the 5 to the next one.
Exactly.
So
That's
kind of like that.
Yeah, exactly. That's correct, right? And a lot of people kind of struggle because they're like, I don't know what the 5 and the 3, I don't understand what they're talking about. I'll show you, okay? You're gonna learn this in biochemistry and you're gonna probably learn it in organic chemistry as well. This is carbon 1, right? This is carbon 2. We don't really denote it, but if you want me to denote it, I can just show you. This is carbon, this is another carbon, this is another carbon, this is carbon 3 by the way, okay? This is carbon 4, and this is carbon 5, okay? You see it, right? 1, 2, 3, 4, 5. So we say that the phosphate is on the 5 end, right? Because that's where carbon 5 is. We talk about OH being the carbon 3 end. So put it like this. So what she's saying is that if I want to add another nucleotide to this, I'm gonna add it from here. You never add nucleotides from the top. You never add from 5. That's why we say that we grow in the 5 to 3 direction. So if this is 5, oops, that's 5, that's 3. I'm going to continuously add more nucleotides on this end. So example would be, if I'm trying to connect it, well, the next one would go right here. It would look like this, right? If I want to draw the next nucleotide, so this is here, I'm going to go P, O, O minus. I'm going to go right here, CH2, C, and we can make this guanine, right? Guanine, okay? CH right here, and we'll go OH right here. We'll go OH here. You see what I did, right? Still carbon 1, carbon 2 is right here, carbon 3 is right here, carbon, oh, that's probably where I got messed up right here. CH2, there we go, right? Carbon 3, carbon 4, and carbon 5, again. So you see my point, right? When you add this, water releases, condensation reaction, because we're bringing two things together. Now, you get it, right? It's pretty self-explanatory,
Yeah.
right? Okay. When you are having DNA, right? DNA is double-stranded. If I have if I have DNA, right? Typically that means that if this is from the 5, if this strand is going from 5 to 3, then if it's connecting to another thing, right? Because remember, this would be adenine on this side, right? And then we would basically draw this structure here, so I'm not going to draw it right now. And then we'd have cytosine on this side, right? Typically saying going this way up would be 5', 3'. See, the whole idea here is that you're going from 5' to 3' for one strand. On the other strand of DNA, right, because there's 2 strands to make the helix, it would be going from 5' to 3'. You want it to be what we call anti-parallel. The reason why is, like, think about it. If 2 things, how I like to think of it is that if 2 things are going in the same direction, it's not that good of attraction. Do you know what I mean? You want things to be going in the opposite direction because it allows for more interactions between your hydrogen bonding. You're gonna have better hydrogen bonding if 2 things are going in the opposite direction, right? Versus if 2 things are going in the same direction, you might have a hard time trying to get those hydrogen bonds to each other, right? 2 hydrogen bonds here, 3 hydrogen bonds here, okay? That's important. Again, why is it important? Like, if I have a lot of CGs in DNA versus a lot of ATs, what would you assume from that?
It would be weak.
If I have a lot of CGs,
it would be strong.
Right?
It has 3 bonds.
exactly.
And
So
it has 2 bonds.
exactly. So if I have C and G, that gives me 3 H bonds, right?
Right.
And then if I have A and T, that gives me 2 hydrogen bonds, okay? It just means interaction. The more bonds you have, the stronger it is, the harder it is to break down, okay? The more of AT you have, it's not that good compared to CG, okay? So the next thing I'm gonna kind of go over, let's see what we get here. Okay, we went over nucleotides, we went over the function, right? We went over how to link them, phosphodiester linkages right here, right? Adding onto the 3' end.
Yeah.
Yeah.
I have a question.
Tell me.
What was the difference between the nucleotide and nucleoside?
So with a nucleotide, you have those 3 components. Phosphate, you got the pentose sugar, which is right here, and then you also have the nitrogen base. The nucleoside has this base, and it also has, sorry, there's somebody at the door going confused. It has the nitrogen base, it has the pentose sugar, and it doesn't have the phosphate. Okay, that's the difference. No phosphate on a nucleoside. Makes
And
sense?
what is nucleoside used for? Like,
I mean, I honestly don't know because we don't really talk about it much. I would just say that they probably found, I'm sure there's a usage for it, I'm just not familiar with it because I spent so much time knowing nucleotides that I haven't really brushed up on it. I asked Dr. Fondufe. Yeah. Now, DNA versus RNA. That's probably something big. You're gonna see this in the future, 100%. At least one question you're gonna get in your lifetime asking this, okay? So, DNA versus RNA, the biggest difference, and you can tell by the actual name. Do you know what DNA stands for?
ribosomal nucleic acid.
Deoxy.
Yeah.
Yeah, deoxyribonucleic acid. And then if I ask you what RNA would be, just ribose, right? Ribonucleic acids. So it's the same idea, right? That nucleotide is right here. Okay. Nitrogen base. I'm going to denote as NB. I'm not going to write out the whole phosphate. I'm just going to write it as PI. It is carbon 2 that you need to watch out for. This is carbon 2, okay? Right? Because it's carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, right? This carbon 2 right here is either going to be an OH or it can be an H. Let me ask you, is it DNA that has the H on carbon 2 or does it have the OH?
OH.
You think DNA has the OH, right? Wrong, okay? So DNA is deoxy. Deoxy.
Okay.
Think about it. Usually, did you You're gonna learn this in organic chemistry, okay? When you have an OH somewhere, we call this a hydroxy group. Hydroxy, right? Hydroxy. O is the oxy, hydro is the H. Deoxy, just non-scientifically speaking, means that there's probably deoxy, there's no OH there. So if it's just the H there, we call this DNA. Okay? RNA is a little bit different. It's gonna be, what do you think? Right, that's it. That's the difference. The other thing that you want to know that for the future, a lot of classes, a lot of teachers will ask you, which one's more stable, DNA or RNA?
DNA.
Why?
Because it has a complete water molecule, or maybe more bonds.
Yeah, I mean, yeah. Generally speaking, it has those H bonding right between those other strands and stuff. Yeah, that can make it a little bit more stable, but it's actually the carbon too, right? When you have this OH in RNA, right, because this is RNA, it's very destabilizing. It's almost as if you're adding an electronegative group, and it's either pushing or pulling things away from each other. It's flipping things around, right? It's not that fun. When you have the H there, though, it provides a little bit more stability, okay? If you think about it, it's kind of small compared to an OH, right? It's not too forceful in any way, okay? That's how I like to think of it. If you want to go learn more scientifically on it, I'd recommend asking Dr. Fondufe. That's just how I like to think about it. Now, here's the other thing though. A lot of people don't realize this, but with RNA, is it single-stranded or is it double-stranded?
Single.
Right? Now let me ask you. We know that DNA is double-stranded, right? Like this, and it can also interact with another bond, or another strand. RNA is a little bit different. RNA, yeah, it's one strand, but that one strand can flip over itself, right? It just does that. You need to know this because in the future, to give you an example, you might see things like this. That's RNA. You see this thing here? It's called a hair loop. It's not good. It just means that you couldn't find anything to bind with. That's just the main thing. You see a lot of RNA in viruses, things like that. And typically the reason why it's able to kind of, I guess, be functional is because of this. It's able to still loop around itself even though there's no two bonds or two strands. So that's that. Okay. I'm gonna hit the central dogma. What's the central dogma?
I'm not sure.
It's okay. I think a lot of people, here's the thing, with cell biology, everyone doesn't realize that this stuff is so important for biochemistry. Like, I can't even begin to tell you that when I was teaching biochem last semester, I would ask somebody, I was like, what's the central dogma? And they'd be like, I don't know what the central dogma is. And I'd be like, you should really know what the central dogma is. Because if you understand what it is, then you can understand why you're memorizing transcription, replication, and translation, okay? So, in your body, you have proteins, right? Every single part of your body makes a different protein. So what I'm saying here is that DNA, in every single cell, is your DNA the same or is it different?
Same.
It's the same, yeah. So your heart cell is gonna have the same DNA as your liver cell. But here's where things would get a little bit different. DNA is the same, but there's a portion of the DNA that codes for different proteins. I'll give you an example. We have a protein in our heart called a neuropeptide hormone, or ANP. I'm not sure if I'm saying it correctly, I can't remember it. But this protein in your heart, okay, is really important for controlling your blood pressure. Now, if we go to your liver, you have an enzyme that does things for glycolysis. You'll learn it later, but maybe I should just tell you right now so you're not like, oh my god. Phosphofructokinase, okay, 1. You're gonna see that specific protein in your liver. You're not gonna find it in your heart though, right? Because think about it, your heart controls blood pressure. Do you think your liver controls the blood pressure? Not really. It's usually breaking down sugar so that you can run away or store sugar as what? How do you store sugar? Glycogen. Glycogen is the storage unit for sugar, okay? That's where you're usually gonna find it, it's your liver. And if you want to break stuff down, you use that enzyme to break it down in your liver. You're not going to find it in your heart. That's the whole point here. But now, that's the point. The DNA is the one that's going to actually code for those proteins. So, if this is my DNA, okay? Let's say I'm in the heart, okay? If I'm in the heart, I'm gonna look at this portion right here, this segment of DNA, okay? This segment of DNA is going to code for a heart protein. Let's write it right here. Heart protein, okay? Now maybe this segment of my DNA, or maybe let's go here. This segment of my DNA can do my liver protein, okay? The whole idea here is that we need to figure out which one we're trying to make. That's why we talk about transcription, translation, to make those proteins, okay? So, the first step of Central Dogma is what? Replication, okay? So with replication, the idea here is that you don't want to use, it's not that you don't want to use the original strand, it's just the fact that you're trying to make a protein, right? And if you want to make as many proteins as you can, you're not going to use the same strand. You're going to try to replicate this strand, okay? So you're going to go through a replication process. Replication. What it's going to do is it's going to make this strand, okay? It's just going to get this strand, okay? And we still have this strand right over here, okay? It replicates it. So now we have the single strand of this DNA, okay? Now, we're going to talk about transcription, okay? Transcription is like this. This is DNA language, right? Remember DNA language? DNA language is, we're talking about A, G, T, and C. If we want to make RNA, okay, and I'm going to explain why we do RNA. RNA language is what? A, G, U, C, okay? Right now this is in DNA language. The body does not know how to translate this. It's like, okay, I'm looking at DNA and I want to make a heart protein, but my heart does not understand it doesn't understand what it's trying to do. So you have to transcribe it. Transcribe, right? You're writing it. You want to write it into RNA so that you're only getting this protein that you want, right? Because you want to write it. So it's going to go through transcription. When I get this transcription, it's only the heart protein I have now. That's all I have. You did not transcribe this whole thing. Remember, this is the one strand of DNA, right? That we replicated. All we did was replicate the DNA, okay? Just one strand of it. When you transcribe it, the body has a mechanism, you'll learn it in genetics, that it's only going to transcribe this one portion that is trying to do the heart protein. That's right here. It transcribed it. It wrote it out for you. Now it's in the form of A, U, G, C, C, G, and maybe U, G, A. Okay? U, G, A. You see what I did? I just wrote whatever it wrote. You remember how to do the opposite base pairing stuff? A to T, C to G? That's basically what happened here. It wrote it out. After transcription, the body's going to say, okay, I understand. I know how to read this. Now let's go make the actual protein. These are the directions for protein, okay? We're going to call it directions. That's the directions. Translation is translating it. Now, I'm going to actually make it. I'm going to make the protein, right? So now we're going to go through translation, okay? And translation is essentially going to be the synthesis, right? We're actually going to make the structure of it. So, what do you think is going to happen here? If we're talking about translation, right? We want to actually make the protein. That's the directions. This is the synthesis. It's the structure part. What do you think?
Sequence of amino acids?
Exactly, right? It's going to make the sequence of amino acids. This might be a lot for you, but if you're taking genetics, why not, okay? So, this part you don't need to know, but I'm just going to show you. You see how there's like AUG, CCG, UGA, right? The body, when it's trying to translate that into amino acids, right? Translation is reading, right? The directions. It's translating the directions to make protein, okay? It's reading this DNA. It's reading these sequence in threes. So AUG, 1. CCG, 2. UGA, 3. Every 3 bases that it reads, we call that a codon, okay? It's coding for an amino acid. AUG codes for methionine, okay? So that will be my first amino acid that I make. And just so you know, methionine is usually the first amino acid that you make in every single part of your body, okay? You start with AOG, that's the start sequence. It says, go ahead, I want you to translate it for me. Then after that, it's gonna read CCG. I don't know what CCG is, but we're just gonna call it amino acid 2, okay, AA2. Now, you start to read, it makes more and more amino acids, okay? Then we might come up to UGA. UGA is a stop codon. That's how it tells you to say, it reads it, it hits UGA, and it's like, okay, stop, you made your heart protein. This is your heart protein, go control the blood pressure now. See what I'm saying? So, ultimately, you take your strand of DNA, you replicate it, right? You just want to get the whole strand, right? To make the process faster. The body has other places, it has other proteins it wants to make. So now, what it's going to say is, okay, I want to get the directions. I don't understand this language. I want to put it in language that I do understand. So we're going to make RNA, okay? Just so you know, have you heard of mRNA? Messenger RNA? Listen, this is technically called mRNA, okay? It's technically called mRNA. You can call it RNA, you can call it mRNA, because at the end of the day, it still has RNA written in it. We call it mRNA because it's messenger, because it's the messenger to make the protein, okay? That's how I think of it. You want to get the directions? Directions say, okay, I'm going to read it, right? I read this thing, and I only print out the directions for the protein that I need. It's right here. Now you got the directions. I want to translate it so I can make the structure. I do translation. You don't need to know the codon part. I just wanted to tell you that because it's cool, right? Starts, stops. Protein ready, it goes out. The last thing I want to ask you is this, is that the cell has different parts of where all of this takes place, okay? It's not just going to happen in one place. Think about it. That would be a little bit messy, in my opinion, to have everything in one part of the cell, okay? It's not. Let me ask you, where is DNA found in the body? In the cell.
Nucleus.
Exactly, right? So you're going to find replication in the nucleus. Nucleus. Transcription. Where do you think transcription happens? Nucleus. All right, it happens in the nucleus. And I'll give you a really good reason why. The reason why it's happening still in the nucleus, well, no, there's more to it, and I'm not going to tell you because that's more of a genetics thing. So, but if you just a non-scientific way to remember this is that you still have DNA, right? And DNA to get into RNA, mRNA, it's still DNA. Like, you haven't fully gotten to RNA, so you're still dealing with DNA. So we're going to say we're still in the nucleus. Okay, that's kind of what I want to think about. Translation is a little bit different. Where do you think translation happens?
Cytoplasm?
Yeah, it happens in the cytoplasm. So the reason why is, think of it like this. You got the directions for the protein in transcription, right? You got the directions. Why do you want to make the structure now? It doesn't make sense to make the protein in the nucleus. What's the nucleus going to do with it? You want to put in the cytoplasm, okay? Because that way it can get sent out to other parts of the cell. Where do you need that protein? Where does it need to go, right? You need to send it out of the nucleus. And in the cytoplasm, do you remember ribosomes?
Yep.
Ribosomes, yeah. Ribosomes are typically found on the rough endoplasmic reticulum, which is in the cytoplasm, okay? It's a structure outside, it's in the cytoplasm. You make the protein on a ribosome. We call the ribosome a protein factory synthesis. It's just a factory, it's in the cytoplasm. After you get it, boom, 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 it's out. You release it, it does its job. That's the central dogma. Central dogma is just trying to make proteins for your body. That's it. And the way to do it is like this. You do the same process if you were in your liver, in your kidneys, right? In your legs, things like that, the skeletal muscle. So I think that's all I have.